Hello, and welcome to a brief introduction to Slicer DMRI. Slicer DMRI is an extension to the program Slicer for medical image visualization and analysis. This extension contains modules which focus primarily on diffusion magnetic resonance imaging, a non-invasive method for mapping white matter connections in the brain. Diffusion MRI, or DMRI, has two main applications. First, tractography to map white matter fiber tracks, and second, imaging of cellular microstructures. This video will be an overview of some of the capabilities of Slicer DMRI, as well as display the available tutorials and resources that can help you learn all there is to know about this extension. This tutorial is aimed primarily at researchers and students just beginning to use Slicer DMRI. However, it is very likely this video will have useful information for anyone interested in Slicer DMRI and its many capabilities. Here is a list of the topics covered in this video. First, we will take a look at some of the capabilities of Slicer DMRI. Some of the footage I'm showing here is from the video tab of the Slicer DMRI website, dmri.slicer.org. After this video, I would strongly recommend going to the website and exploring what it has to offer. Here is an example of a use of the Tractography display module. Using the sample DTI brain found in Slicer's sample data, we can examine the fiber bundles and their paths in this brain. And here we can see Slicer DMRI's potential use in neurosurgical planning. Using this software, it's straightforward to image the tracks in respect to tumors and other structures in the brain. It's even possible to perform measurements which show the exact distance between these tracks and the tumor itself. A less visual but equally important part of Slicer DMRI is its ability to convert one file format to another, and here we can see the module with that specific ability. Next, going back to our list of topics, I'm quickly going to show you how to download Slicer DMRI from the extension manager. As you can see, I'm using Slicer version 4.6.2. Don't worry if you're not using this version or you are on Linux or Windows. The process should be exactly the same. First, we must navigate to the extension manager, which is the blue icon on the right side of the top bar. And now we can search for Slicer DMRI. After clicking download, we wait for the installation and then restart Slicer when it finishes up. To make sure that Slicer DMRI was installed correctly, we can take a look to make sure that the modules that are part of this extension were added. Next, we will talk about each of the tutorials that connect with Slicer DMRI. They can all be found on dmri.slicer.org. We will summarize what you can learn from each tutorial. If one interests you, please head to our website where you can find these tutorials and many other resources where you can learn more about Slicer and Slicer DMRI. The first tutorial listed on our website is a diffusion weighted image converter tutorial. This short tutorial teaches how to use the DWI convert module to convert between file formats. The Slicer Diffusion Tensor Imaging tutorial is broad. It covers a wide range of topics and modules and is a great introduction to Slicer DMRI's visualization abilities. The next tutorial is called the Slicer Neurosurgical Planning tutorial. Using sample data in this tutorial, you will learn how to use the tractography seeding modules alongside the modules included in Slicer to explore the white matter fibers surrounding a tumor. In the Slicer Unscented Kalman Filter Tractography tutorial, you'll be using many of the same modules as you would in the Slicer Diffusion Tensor Imaging tutorial, although with the goal of the results shown here, which is a tractography of the corpus callosum using UKF. The next tutorial, entitled Fiber Bundle Selection and Scalar Measurements, is a follow-up to the Slicer Diffusion Tensor Imaging tutorial. It builds on many of the concepts presented in that tutorial and has the goal to use a whole brain tractography for fiber bundle selection and scalar measurements. The final tutorial, Command Line Interface Application, has the goal of teaching users to run Slicer commands from the command line rather than using the graphical user interface. The purpose of this technique is to perform batch processing for scientific studies using data from multiple subjects. And this concludes the tutorial section of this video. However, these tutorials are just the start of what you can learn about SlicerDMRI. We also have links specifically for those who are interested in the development side of SlicerDMRI, including an example short tutorial from the Slicer Wiki script repository, as well as a link to a GitHub repository that has SlicerDMRI's source code. Also located on the SlicerDMRI website is a link to the ever-growing Slicer forum, where you can ask any questions you may have about SlicerDMRI or Slicer in general. Finally, in the Citations tab, you can take a look at some of the publications that cite SlicerDMRI. And that is all for this video. I hope it was helpful for you, and thank you so much for watching. Please visit our website, dmri.slicer.org. In the description of this video, you'll find a link to the front page of our site. There you will find the latest on our extension. Thanks again.